Les damos a todos la más grande bienvenida a otro coloquio de microbiología de Valparaíso. Hoy día tenemos una excelente amiga y que la conozco hace mucho tiempo. Ella se llama Macarena Marín. Es una chilena que trabaja en Alemania. Ella, les voy a contar un poquito su currículum. Ella estudió ingeniería en biotecnología en la Universidad de Chile y después decidió irse a Alemania, a la Universidad de Braunschweig, para sacar el doctorado en ciencias naturales. Y ahí es donde nos conocimos, porque coincidió que llegó al mismo laboratorio donde yo estaba haciendo mi doctorado. Y fue realmente genial porque ella continuó el trabajo que yo estaba terminando. Ella continuó con el trabajo en el mismo grupo. Y este grupo es del doctor Dietmar Pipa, que también le tenemos mucho cariño. Y después ella decidió quedarse en Alemania y se fue a realizar el postdoc con Thomas Ott en la Universidad de München, que también se conoce por LMU, que la verdad es una de las 3 mejores universidades ranqueadas en Alemania. O sea, es una universidad muy con alto prestigio. Y ella estuvo ahí casi 5 años trabajando más bien con plantas, el lado oscuro, no tanto con bacterias, pero adquirió mucho experiencia con las plantas. Y en el año 2015 decidió formar su propio grupo independiente bajo el alero de Martin Paniske. Y ahora lo que ella trabaja es la interacción planta-bacteria, a veces trabajando por el lado de las plantas y a veces trabajando por el lado de las bacterias. Entonces, muchas gracias por aceptar nuestra invitación, Macarena, y por supuesto que te dejo para que presentes.
Súper. Muchas gracias, Bea, por la introducción y la invitación a participar en este coloquio. Como la Bea dijo, hoy voy a contarles sobre la investigación que mi grupo hace en Múnich. Esta se centra principalmente en cómo bacterias fijadoras de nitrógeno establecen una relación simbiótica con plantas de la familia de las leguminosas. En particular, yo estoy interesada en cómo las bacterias infectan a la planta y cómo la planta crea un ambiente propicio para la fijación de nitrógeno. Esto ocurre en el interior de células especializadas en las raíces de la planta. Como pueden ver en esta micrografía electrónica, la planta acomoda las bacterias en su citoplasma, pero las rodea con una membrana plasmática. La pregunta es cómo las bacterias son capaces de cruzar la pared celular de la planta sin perturbar la integridad de las células hospederas. Pero antes de contarles más sobre este proceso, me gustaría ponerlo en un contexto un poco más global y a partir de ahora voy a cambiar al inglés para el resto de la presentación y espero que no haya ningún problema. So I will start by reminding you about the importance of nitrogen. So nitrogen is essential to support life on the planet. It is a central component of biomolecules such as nucleic acids, proteins, and chlorophyll. Although nitrogen is highly abundant in our atmosphere, plants, animals, and eukaryotes in general cannot absorb it directly from the air, so they rely on other sources. Plants, for example, can absorb it directly from the soil. However, there it can become scarce and tremendously limiting for plant growth. In these images, you can see symptoms of nitrogen deficiency. So this is, for example, stunted plant growth and leaf chlorosis. This is this characteristic yellowing of the leaves. And of course, this transduces to dramatic losses in yield. So to prevent this, farmers apply nitrogen fertilizers. However, these come at a great economical and environmental cost. Since the invention of the Haber-Bosch process, which is the major source of synthetic nitrogen fertilizers, we have seen the biggest demographic explosion registered so far. It has been estimated that currently roughly half of the human population is fed by crops fertilized with synthetic nitrogen fertilizers coming from the Haber-Bosch process. However, this at the same time has created a complete imbalance in the nitrogen cycle in the planet. The importance of this issue is highlighted in this graph that illustrates the planetary boundaries that we should not cross if we want to keep the stability of the Holocene. Here you can see how the boundary for the nitrogen cycle has been completely overstepped. I think this is best illustrated with the problem of eutrophication, which causes massive losses in biodiversity in aquatic ecosystems. The cause of this is the leakage of nitrogen or phosphorus into water sources that lead to the uncontrolled growth of algae and the creation of anoxic zones. Wait a second. So we are confronted with this dichotomy of keeping up with agricultural production and the population growth while maintaining healthy ecosystems. And this is not only an issue that concerns farmers or environmentalists. This is absolutely essential for every one of us. And I would, of course, be delighted to discuss more about this during the question session. This issue is, of course, extremely complex and cannot be solved only with technical solutions, as some people want to believe. It requires changes in consumer behaviors, policy making, and of course, more research. So what can we do from the side of research? So what can we do, for example, as a scientist? I believe that when you don't know where to start, always look at nature and see what solutions has it already created. So the natural way of making nitrogen fertilizers is by a beautiful multiproteic enzyme produced by some bacteria and archaea called the nitrogenase. This enzyme catalyzes the reduction of this very stable triple bond in the dinitrogen molecule to form ammonia. Some nitrogen-fixing bacteria even establish symbiotic associations with plants and thus help them overcome nitrogen limitation in the soil. So if used properly, these associations could constitute a sustainable alternative to synthetic nitrogen fertilizers. So these solutions are very varied and range from optimizing already existing crops that associate with nitrogen-fixing bacteria to using these plants in rotations with non-symbiotic crops to more intricate solutions, for example, engineering the symbiosis in non-symbiotic plants or even transferring directly the nitrogenase to the host. Of course, all of these solutions require a deep molecular understanding for this symbiosis. So now I will tell you a bit more about this molecular basis of this symbiosis. So this capacity of associating with nitrogen fixers is spread across a wide range of land plant taxa, as shown in this phylogenetic tree. This impressive plant diversity is mirrored by a diversity on the side of bacteria, which includes cyanobacteria, filamentous gram-positive, frankia, and rhizobia, a polyphyletic group composed by alpha and beta proteobacteria. In the majority of these symbiotic associations, bacteria are hosted outside of plant cells, with the exception of Cunera, that it's the plant of the nalca leaf that we use in Chile to make curanto, and plants from the orders Fabales, Fagales, Rosales, and Curcubitales, here indicated or highlighted in red. This group of plants here in red have evolved an endosymbiosis where they host bacteria inside of cells of specialized root organs called nodules. And today, for simplicity, I will talk exclusively about the symbiosis between Fabaceae, a family within the Fabales, and Rhizobia. These plants, these Fabaceae plants, are also known as legumes and are of great importance in agriculture. This is because members include grains such as soybean, peas, peanuts, and forage plants such as alfalfa. The most characteristic feature is the formation of these nodule structures in the roots. These nodules are like tiny nitrogen fixing factories that are absolutely packed with bacteria. And so it's within these cells that that nitrogen fixation takes place. So nitrogen fixation is a very costly process in terms of energy, as it consumes at least 16 ATP molecules per single dinitrogen molecule. So to fuel this reaction, the plant allocates, in some cases, up to 30% of its photosynthetic products to the nodules to feed this rhizobia. So the establishment of this symbiosis supposes a huge energetic commitment and an inherent risk of attracting, for example, pathogens and parasitic rhizobia inhabiting the rhizosphere. Therefore, the control of who enters these nodule cells is of critical importance. This opened one of the very central questions in the root nodule symbiosis field, and this is how legumes select rhizobia from complex microbial communities. So the plant imposes a very stringent filter that is clearly seen in this study from the labs of Simona Radu-Toiu and Paul Schulze-Leffert, where they investigated the alpha diversity of bacteria colonizing the soil, the rhizosphere, the root and nodules from a legume plant called Lotus japonicus. And as you can see in this graph, the diversity of bacteria in these compartments dramatically decreases, especially in the nodules, which are compartments dominantly colonized by rhizobia. So how does the plant achieve this very stringent filter? Under nitrogen starvation, the roots of legumes exude flavonoids that have a dual function. On the one hand, they attract rhizobia towards the root, and on the other, they induce the biosynthesis and the secretion of lipoketoligosaccharides, or so-called nodulation factors. These molecules are then recognized specifically by receptors located at the plasma membrane of the host, and perception of these molecules and activation of these receptors activates a complex signaling pathway that culminates in transcriptional reprogramming of cells in the root of the host. Today, I don't have the time to go in all of the details, but the bottom line is that the signaling pathway activates in the host two distinct but interconnected processes. On the one hand, cortical cell divisions that lead to the formation of this organ, the nodule, and cellular rearrangements that lead to the internalization of bacteria inside of plant cells, the so-called infection. The challenge is how bacteria can go from this rhizosphere state to reach specific cells inside of the nodule. For this, they need to cross multiple cell layers in the root, but they cannot simply make a hole in the root because this would risk eliciting stress responses in the plant. So therefore, they rely on this infection process that is highly complex, and as it's central to my work, I will spend some extra time explaining a bit about it. So different infection mechanisms have been described depending on the host and the bacteria. So in the best studied mechanism, the host entraps bacteria in root hairs, that here you can see in this micrograph. This is a root hair in the plant, and this you have bacteria labeled with a green fluorescent protein. And you can see how the root hair curls around the bacteria and entraps them. Then the plant creates some tubular structures called infection threads that guide the bacteria through different cell layers. And this is indicated by this red arrowhead here. I hope you can see this kind of tube that the plant forms. This is illustrated in this scheme, where you can see that the tip of the infection thread reaches the boundary between two cells that, for example, could happen here or here. And while this happenings, at the end of the receiving cell, already the cytoplasm starts rearranging to make space for the upcoming, or for make space for a new infection threat that will develop here. And this process repeats itself until the bacteria reach specific cells in the nodule where they are internalized. In some tropical legumes, bacteria penetrate the epidermis by natural openings in the root, for example, at sites of lateral root emergence, as you can see in these pictures, where bacteria are colonizing an emerging nodule at the base of a lateral root. So this creates little cracks through which bacteria can enter. And this is very characteristic of plants such as Spania rostrata. In others, they can go simply through epidermal cells. So, however, in all of them, the end product is the same. Bacteria enter dividing cells in the nodule primordium. They are surrounded by a plant-derived membrane, and they differentiate into nitrogen-fixing bacteroids. And even though this step of the entry into the plant cell is one of the most critical steps of the symbiosis, what we know about it, it's mostly limited to descriptive microscopical observations, and we don't know who are the key molecular players controlling the remodeling, for example, of this interface to allow the crossing of the bacteria crossing of the cell wall without bursting the cell, and of course all of the later metabolic and cell biological adaptations that the host cell needs to undergo to host the rhizobia. So to understand this at a mechanistic level would be absolutely fundamental if we ever want to transfer, for example, this symbiosis to non-legume plants. So in my group, we are interested in understanding these two processes. So one, how rhizobia enter the plant cell, and second, what it's like the right environment, or how the plant creates a right environment so that the bacteria can differentiate into these nitrogen-fixing bacteroids. So what are all of the changes that the cell needs to undergo to do this? And these, of course, as you can imagine, these are very complex questions that, in my opinion, can only be solved if we study them from the plant and the microbial side. That is why in my lab, we are establishing methods to study both partners. Some of these we use routinely, like generation of mutant strains, bacterial genome sequencing, profiling of bacterial communities, Golden Gate cloning, or confocal microscopy. Others we are still setting up, and we are and they are underway at the moment. And this is, for example, CRISPR and transposon sequencing. Today, I will give you a sneak peek into two projects, one from the side of the bacteria and one from the side of the plant. So in the first project, we investigate how rhizobia transition from this free living state in the rhizosphere to a symbiotic states, so inside of the plant cell, and what are the conditions that are required so that this switch occurs efficiently, so that once the bacteria enter the plant cell, they can start fixing nitrogen. So for this, we use several rhizobial strains that have different phenotypes. However, for brevity, I will show you the results for two strains that have been isolated from lotus plants, so our the plant that we usually use in our lab. One of these strains is beneficial and promotes the growth of its host, Lotus japonicus. The other, and this you can see in this picture by these elongated shoots. The other is parasitic, and So this means that it can induce nodules, but these do not fix nitrogen. And this you can clearly see in these stunted growth phenotypes of plants nodulated with this strain. So the first thing that we did was to sequence the genome of the non-fixing strain, this Rhizobium leguminosarum Norway, that's the name of the strain, and see if it contained the symbiotic plasmid and the NIF and fix genes that are required for nitrogen fixation. We also compared, and so you can see that this plant doesn't promote the growth of plants. And we also compare it to a closely related strain that can fix nitrogen in P. And this is this other strain, this 3841. So we observed that the subunits of the nitrogenase are highly conserved between both strains. However, a noteworthy difference was that the non-fixing strain harbors a single fixed NOQP operon encoding an essential terminal oxidase, whereas the fixing strain, this 3841, contains 2 copies of this operon. Furthermore, the Norway strain, so the non-fixing strain, lacks genes encoding for the fix K and fix L that are transcriptional regulators. These, together with the FNRN protein, control the expression of the nitrogen fixation genes in other rhizobia. So instead, the non-fixing strain harbors 2 putative FNRN genes. However, this is reminiscent of another strain called UPM 791, where this FNRN is the global regulator of the fixed genes. So these results suggest that the absence of nitrogen fixation is not caused by a lack of the nitrogenase enzyme, but more likely due to changes in the regulation of its expression. So to investigate this, we are currently establishing a live cell microscopy system with which we can follow infection and gene regulation in the bacteria and in the plant. So basically what we want to do is, or actually what we are doing at the moment, is to grow young seedlings on plates under axenic conditions, and we cover them with a gas permeable foil. This allows us to look at the roots with minimal perturbation and follow them, follow the growth, and we can even see and follow the presence of single bacteria that here are tagged with an M-cherry fluorophore, and you can see them colonizing the root hairs of the lotus plants. So the idea is combine this with plant lines carrying reporter fusions of plant genes fused to fluorophores that activate at different stages of the symbiosis. This is, for example, a line. This DR5-GFP NLS is a line created in the lab of Masayoshi Kawaguchi, carrying a fusion between a GFP fluorophore fused to a nuclear localization signal and an OXIM reporter called DR5. And these label early infection sites. And this you can see here by the glowing of the plant cell nucleus here in green on the plant roots. In red, and in these figures in red and in cyan, you can see 2 different bacteria that carry plasmids expressing fluorophores constitutively. So now we can follow infection of these bacteria while they are colonizing the root. And as you can see in these images, the colonization pattern of these 2 strains is completely different at the sites of infection. So this strain can make infection threads, and the bacteria are mostly constrained to these thread structures, whereas the other strain can colonize very homogeneously different, like covering the whole root. We are also generating bacterial reporters so we can follow the transcription of key genes at a single cell level. For this, we use transcriptional fusions of the translational fusions of the first amino acids of the encoded proteins and fluorophores under the control of the native promoters. And with this, we hope that we will be able to resolve expression at a spatial and temporal with spatial and temporal resolution, and we can move away from qRT-PCRs that only show like a more global picture but don't give you this this resolution at a cell level. And the idea at the end is to have a system where we can follow the expression of specific genes, of specific bacterial genes during infection, and see if different treatments or maybe different plant mutant backgrounds can change the expression of these genes. In the case, for example, of our non-fixing strain, we would like to see if there is any condition where we can make it fix. So now I will switch a bit gears and move to the plant project, where we ask a different question, and this is what genes from the plant side control the entry of bacteria into plant cells, and what are all of the cell adaptations to host the bacteria. And these, as I told you before, are fundamental questions, but we don't know very much about them, and you may wonder why this is the case, if these are so important questions, why people have not found the answer yet. So a lot of what we know about this symbiosis is product of forward genetic screens. In these screens, if you are not familiar, plants are basically mutagenized and the progeny is screened for phenotypes. And these phenotypes are usually very striking. For example, loss of nodulation or loss of infection threats. And so once you identify plants that have a phenotype, you can map by crosses, for example, or other methods, you can identify the genes that cause this phenotype. And this approach has been incredibly successful in finding the key components in the root nodule symbiosis. This is, for example, how genes involve in the early molecular dialogue, for example, these receptors, genes involved in early stages of infection or in nodule organogenesis were originally identified. However, the question of how the bacteria enter the plant cell and all of these later cell adaptations is much more complex. For example, mutations that interrupt nodule formation or early stages of the symbiosis block later cell infection. So of course, if there is no nodule, there cannot be nodule cell infection. But this does not mean that the interrupted genes are directly involved in the process. So also, it is likely that some of the genes that control this process are redundant and may even play housekeeping functions. And of course, these type of genes are very difficult to find in genetic screens that rely in very strong phenotypes. Another approach that has been widely used in the field is transcriptomics. However, if you are familiar with transcriptomic experiments, they often yield many candidates and are very difficult, and you end up with so many candidates that at the end it's almost impossible to further investigate these genes at a molecular level. So none of these two approaches is actually optimal to address our questions. So in my lab, we decided to take an alternative approach where we combine transcriptomics, but with the natural diversity of different lotus succession. And I will explain you in a second why we think this approach is more suitable to answer this question. So the original idea was to see if in different closely related plants or different closely related accessions, so these are like types of lotus plants, we could arrest the symbiosis at different steps. And this is illustrated in this scheme here on the top. The hope was that by combining multiple accessions that would be arrested at these different stages, we could minimize accession-specific differences and purify stage-specific ones. then using comparative transcriptomics, we hope to identify a discrete set of candidates that we could validate using different methods. So a bit the concept is like combining genetic diversity or like natural diversity underlined by genetic diversity with transcriptomics. The system that we used was based on different Lotus japonicus accession that formed nodules with different bacteria. And this is illustrated here. So on the top is the name of the different lotus plants, and here on the side is the different strains that we use to inoculate. And these are just pictures of how these nodules look on the root of the plant. So, and what we observe is that certain bacteria would induce nodule formation, but these nodules would be arrested at different stages of the development. And here I just show you a subset of the combinations of plant and bacterial genotypes that we use. So what was quite curious is that this diversity was not evident with a classical symbiont of lotus like Mesorhizobium. So you can see here all Mesorhizobium induced the same nodule phenotype in all plants, but only we could see it when we infected these plants with somehow suboptimal strains that can induce nodules but not fix nitrogen. And we use one of these strains, our favorite strain, this Rhizobium leguminosarum Norway, because it gave a very nice range of different phenotypes. If you can see here, in some plants, it didn't make nodules, while in other, the formation of the nodule arrested at early stages, and in others form completely developed nodules, but these nodules did not fix nitrogen. So to see if we could exploit this diversity on the plant side, we looked at how the bacteria could infect these nodules. So, and just again, a brief reminder about this strain. So this strain was originally isolated from nodules of lotus plants, and it can, but it cannot, but these nodules do not fix. So when we followed its infection, we observed that contrary to Mesorhizobia that infect nodules via infection threads, this strain often accumulated at the top of nodules and penetrated the epidermis directly through what looked like disrupted plant cells. And maybe if you are not familiar so much with microscopy, what you are seeing here, these are roots of the plant that have been sectioned and the cell wall has been counterstarred with a dye that is called calculofluoro white. And the bacteria that are infected are transformed with a fluorescent protein that is expressed constitutively, and here are indicated in green. So, and then we could see that these two different strains had different infection mechanisms. But another very important difference between these two strains was that the Mesorhizobium, that in contrast to Mesorhizobium, this Rhizobium leguminosarum strain, in this strain, the infection was not in sync with organogenesis. And what do I mean with this? So as you can see here, Mesorhizobium, the cells in the inner tissue of the root are being infected at the same time that they are dividing. So you can see formation of the nodule and infection of the cells. In contrast, in this with this other strain, you see formation of the nodule, but the cells at the early stages remain uninfected. So we have like this connection between the infection process and the organogenesis. However, at later stages, bacteria were internalized and symbiosomes, so these structures inside of the plant cell, were formed. And this you can see in this electron microscopic picture, where you see plant cells. Here are, for example, cells that are empty, so they are not infected. And here you have plant cells that are filled with bacteria, here in these symbiosomes, and here you see like a plant nucleus. So we had a very promising system. So now we could go back to our original question of what is the infection phenotype of these different accessions, and see if we could find accessions that would make nodules but would not be infected. Based on a preliminary screen, we selected 30 Lotus japonicus accessions that seem to have different colonization levels. However, none of these accessions developed infection threats. But what was more exciting, though, was that in some accessions, the majority of the nodules were infected, while in others, only few were colonized. And this you can see in the percentage of nodule infected in these different accessions. There was also a correlation with the percentage of nodules infected and the colonization degree that you can see also in this section of a nodule. And here you can see where nodules from these accessions tend to be more colonized than nodules from these other accessions that form few colonized nodules. And this was very exciting, as the overall nodule morphology between the different accessions was roughly the same, and they mostly differed only if they were infected or not. So we had an ideal condition to compare differences in infection and in accommodation, disconnected from differences in organogenesis or fixation. So what my PhD student Rafa did was to grow many plants from these different accessions and screen them under the microscope, harvest primordia and infected non-infected nodules. And so the rationale was to compare these infected and non-infected nodules to identify accommodation candidates, so the genes that are involved in hosting the bacteria, and uninfected primordia from accessions that later become infected or accessions that remain uninfected, to identify genes that may mediate entry. Also, we looked at nodules that were infected with Mesorhizobium loti to identify differences between nodules that fix nitrogen and nodules that do not fix nitrogen. So in total, we had 7 different Lotus japonicus accessions with 6 biological replicates per condition, yielding a total of 78 RNA-seq libraries. So we proceed with our pipeline, doing RNA sequencing. And if you imagine the 78 libraries, it's a lot. And we were able to afford for such a large scale experiment because our next door neighbor, Wolfgang Ennard, had developed a methodology called SCRB-seq. That allows you to create a large number of RNA-seq libraries at a very low cost. And this methodology was originally developed for single-cell RNA-seq approaches, but we adapted it to our nodule samples. So the first feature of this method is that the cDNA libraries are constructed using molecular crowding that increases the sensitivity over conventional library preparation methods. And here polyethylene glycol is thought to reduce the effective reaction volume and thereby increases the cDNA synthesis efficiency. This, of course, confers high sensitivity. The second feature is that it uses unique molecular identifiers, or UMIs, to remove amplification biases and to allow multiplexing of many samples. So And the last thing is that in addition, it only sequenced from the 3' end of the transcript. So it only sequenced the 3' end of the transcript. So, and all of these things in combination give high accuracy for the quantification of the expression levels. So then Rafa conducted a principal component analysis, and you can see clearly in this PCA plot how the different samples cluster according to the different categories. So in this corner here, you can see samples that come from nodule primordia, so either nodules that will become infected or non-infected. Here samples from non-infected nodules, here samples from infected nodules, and here very far away are the samples that come from fixing nodules. So these gave ideal conditions to do differential gene expression analysis to identify candidate genes. Using this approach, so far we have identified 50 candidates for entry and around 150 genes for accommodation. But what is super encouraging is that we not only found some novel genes, but we also found genes that are known to play roles in these different processes. So these are our kind of proof of principle genes. And these, just some examples of these are genes encoding for arabinogalactan and extensine proteins, and also like genes encoding for APN1 or for the sulfate transporter SST1. So just to give you a brief glimpse, so for the candidates potentially involved in entry, we found several genes involved in signaling and gene expression, lipid metabolism, organelle functioning, cytoskeleton, and cell wall remodelings. These are all functions that seem quite plausible for controlling the entry of bacteria. In contrast, for the accommodation candidates, we found completely different set of genes. So we also found some signaling candidates, but predominantly we found genes involved in transport and metabolism. And we also found some genes involved in oxygen, homeostasis, and suberin and lignin, suberin biosynthesis and lignin deposition. So oxygen homeostasis in the nodule is critical for efficient nitrogen fixation, as oxygen inactivates the nitrogenase enzyme, but low oxygen tensions are absolutely necessary to ensure mitochondrial respiration. So in order to maintain nitrogen fixation, the nodule has several structural and metabolic adaptations. So the structural responses are, for example, the creation of a physical barrier that limits oxygen diffusion, while the metabolic responses include binding of oxygen by a protein called leghemoglobin. What was really interesting in our data set was that we observed that genes encoding leghemoglobins, these are among this category of oxygen homeostasis, co-express specifically with genes involved in suberin production and lignin deposition. But what is suberin and lignin, and why this is interesting? So suberin is a complex cell wall polyester composed by long-chain fatty acids that is highly hydrophobic. So in plant roots, suberin is deposited in cell walls of the endodermis that you can see here in this scheme. And this is the cell layer in the root that prevents the apoplastic movement from molecules into the vasculature. It also prevents pathogens to reach the vasculature of the root. And this is aided by the so-called Casparian strips that are made of lignin. So we ask ourselves if it could be that suberin and lignin genes, so that the suberin and lignin genes that we found in our data sets and that are co-expressed with leg hemoglobin could mediate cell wall modifications in this layer that limit oxygen diffusion into the nodules. And so far, just to specify, these genes are not known. So the genes that mediate this process, the formation of this structure, are not known. So what we have done is to stain nodules, again, nodule sections. So you can see an image here with suberin and lignin stainings. This can be seen in this confocal microscopic picture of a nodule section stained with basic fuchsin that stains lignin. You can see in the staining the root endodermis surrounding the vasculature, and also here in the nodule vasculature. However, in addition, we see an extra-cell layer here that is also stained, that it's outside or surrounding the vasculature. And we hypothesize now that some of the genes that we have identified in our transcriptomics may mediate the production of or may aid in the formation of these cell wall modifications in this cell layer. So we could, and just one thing that I forgot to mention is that so these genes are specifically upregulated in the nodules that are infected, and comparatively downregulated in nodules that are non-infected. So we could envision that once bacteria, for example, colonize the inner tissue of the nodules, these genes are upregulated and mediate cell wall modifications that seal the nodule, for example, limiting oxygen diffusion or secondary pathogen infections. And of course, in uninfected nodules, as bacteria do not colonize, this probably does not occur or occurs at a lower level or it's delayed. So we are characterizing these genes molecularly at the moment to test this hypothesis. However, the big limitation that we find is that all of the genes that we find belong to complex multi-gene families. And here I just show you the phylogeny for one group of candidates. So among our genes, we found several genes belonging to the CASP or CASP-L family involved in the formation of Casparian strips. So there are many genes belonging to this family in lotus, and several of them are specifically upregulated in nodules. In our data set, even we found several genes from this subfamily 4 indicated in green. So to investigate the challenge is that we need to be able to silence all of these genes simultaneously. And for this, we are using artificial microRNAs to see if we can then see phenotype. But as you can imagine, like working with so many genes in parallel is not so easy. And once we identified which of these genes are the key players, we want to create CRISPR lines so we can targetedly delete them. So to sum up, the main points of my presentation today, I showed you that synthetic nitrogen fertilization is widely applied, but it comes at a great cost. However, natural alternatives exist, and one of them is this nitrogenase enzyme produced by archaea and some bacteria. Luckily for us, some of these nitrogen-fixing bacteria establish symbiosis with plants, and this is widespread across different land plant taxa. One example of this is the endosymbiosis between legumes and rhizobia, and one of the critical steps of this symbiosis is the infection of nodule cells. And once rhizobia infect these cells, they differentiate into nitrogen-fixing bacteroids if the cell provides a suitable environment. So I would like to finish by thanking my lovely group, who are the ones that do all of this work, especially Yuan Liang, that it's my first PhD student and who is who is about to graduate now, and she did all of this initial characterization of this Norway strain. Also, special thanks to Rafa, who is the student leading now the plant project, and Sarah, my postdoc, that is leading the bacterial project on lifestyle imaging. Hopefully next time I can tell you about the work that Yen, that it's here, is doing on characterizing some plant proteins, and Duncan, that is investigating the microbiome of nodules and the impact that this has on nitrogen fixation efficiency. Also, I would like to thank a large list of collaborators with whom we could have not done this work, especially Wolfgang and Lucas, who did all of the libraries, helped a lot with the library preparation for the RNA-seq, and of course Martin Parniske, who has hosted our lab in the last years. And with this, I'm looking forward to your questions.
Thank you very much, Macarena. That was really, really nice. Perdón. Muchas gracias, Macarena. Estuvo genial tu charla, muy interesante. Me encantaron las micrografías con los nódulos. Muy bello. Quiero preguntar a la audiencia si tienen algunas preguntas para Macarena. Por favor, pueden preguntar en español. La Macarena, por supuesto, que puede contestar en español. Así que, ¿hay alguien que quiere hacer preguntas?
Sí, yo, hola.
Adelante, Esli.
Hola, ¿qué tal, Macarena? Me encantó la presentación.
Muchas gracias.
Voy a preguntar algo que, no sé, me está rondando en la cabeza y veo que usted trabaja en el tema de interacción microorganismo-planta y entonces, no sé si lo han trabajado, pero es algo que me interesa y entonces quizás tienen experiencia en eso. ¿Algunas de las bacterias que ustedes han trabajado induce alguna modificación en las plantas?
¿Puedes especificar un poco qué tipo de modificación?
A ver, hay bacterias o algún otro microorganismo que producen ciertos metabolitos secundarios que inducen resistencia sistémica en la planta. Entonces, cuando ya no hay presencia del microorganismo, la planta aún así es tolerante a patógenos. Este tipo de modificación es la que principalmente, no sé, si tienen experiencia, me gustaría saber.
Yo no tengo experiencia en Systemic Acquired Resistance y en verdad no sé si alguien ha investigado si los rhizobios inducen SAR en las plantas. En particular, o sea, yo no te puedo decir las cepas, o sea, las bacterias con las que nosotros trabajamos no hemos investigado eso.
Ya. No, es algo que he estado leyendo estos días y entonces me resultó la pregunta y digo, bueno, vi la presentación y vi que era el tema bacteria, planta y digo, bueno, a lo mejor quizás ellos tienen experiencia en esto. Está bien, gracias.
Lo que sí estas bacterias hacen, tienen, o sea, se sabe que, o sea, hay evidencia de que estas moléculas, que no sé si te acuerdas de una parte de la presentación de estos factores de nodulación, estos NOD factors,
sí.
Lo que hacen es, hay evidencia de que pueden suprimir la respuesta inmune de la planta, pero no es por Systemic Acquired Resistance, so lo que hacen es suprimir el Pump Triggered Immunity, so PTI. So, hay una conexión entre rhizobios y supresión del sistema inmune de la planta, pero no sé si hay conexión entre rhizobios y SAR.
Ya, gracias.
Muchas gracias. ¿Alguien más quiere hacer una pregunta? Mijael, adelante.
Macarena, súper interesante tu presentación, ¿no? Y te quería preguntar por estas dos cepas, ¿cierto? que si bien las dos tienen los genes de nitrogenasa y tienen casi todos los genes para fijar nitrógeno, ¿cierto? Una tiene la capacidad de fijar nitrógeno y la otra no. Y tú hablaste un poco de que habían dos factores regulatorios involucrados en eso que faltaban en una de las cepas, pero quizás nos puedes contar un poco más eso, digamos, cuán relevante es la regulación en estos procesos, porque en una primera aproximación uno podría ingenuamente pensar solamente en los genes, ¿cierto? Pero tú dices que la regulación es fundamental. No sé si puedes comentarnos un poco más con más detalle esas diferencias.
Claro. O sea, la regulación es súper importante, porque si uno se imagina, la nitrogenasa es una enzima muy compleja y implica un costo muy grande para la bacteria producir la nitrogenasa. Entonces, la producción de nitrogenasa se induce solo en condiciones microaerofílicas. Entonces, está fuertemente, al menos en los rhizobios que yo investigo, está altamente controlada por la tensión de oxígeno. Entonces, solo una vez que la bacteria entra al nódulo y la tensión de oxígeno baja, ahí se inducen los genes para la fijación de nitrógeno. Entonces, el control de la inducción es absolutamente esencial, porque si las bacterias lo indujeran en estado de vía libre, la enzima se inactiva por oxígeno. Entonces, la regulación es crítica. Lo que todavía no sabemos es que la regulación es muy diversa en distintas bacterias. O sea, hay bacterias que usan estos genes que nombré, y hay otras que usan sistemas de regulación completamente distintos. Entonces, no podemos decir porque faltan estos genes que la bacteria no va, que la regulación no funciona. Lo que sí vemos es que la bacteria, aunque colonice los nódulos, no induce la nitrogenasa. Entonces hay algo que está fallando, lo que pasa es que no sabemos por qué no induce la nitrogenasa. Puede ser porque hay un factor que falta y no puede inducir la nitrogenasa, o le falta una señal de la planta, o hay un problema de comunicación con la planta, y aunque tenga todos los genes, no lo puede inducir. Y eso es lo que queremos ver con estos ensayos de live cell imaging.
Sí, pero tú piensas que en ese proceso, digamos, también puede ser que a la cepa que no es capaz de fijar nitrógeno también no tenga suficiente defensa frente al oxígeno. Puede ser que también parte del metabolismo del oxígeno no está preparado para que la nitrogenasa funcione, o sea que la bacteria no la enciende porque eso le podría causar problemas.
Podría ser. Definitivamente podría ser que hay algún problema, pero esta cepa la aislaron de un nódulo que entonces me imagino que la bacteria está adaptada a hacer esta transición de estado de vida libre a vivir dentro de un nódulo con baja tensión de oxígeno. Lo único que lo particular es que esta cepa no estaba sola dentro de ese nódulo, o sea, había otra bacteria que estaba coinfectando ese nódulo. Entonces puede haber algo que la otra bacteria estaba haciendo para crear un ambiente propicio para esta otra bacteria que crece. Entonces puede haber que hay un tipo de interacción no solo con la planta, pero con otras bacterias otras bacterias en el nódulo. Y eso también estamos tratando de ver cuáles son las interacciones entre las distintas bacterias.
Perfecto. Muchas gracias, Macarena.
Muchas gracias. ¿Alguien más le gustaría hacer alguna pregunta? Carolina, por favor.
Hola, buenas tardes. Muchas gracias, Macarena. Muy interesante tu presentación. Y a mí también me gustan los rhizobios. Yo los estudio con la interacción con plantas con fines de remediación de suelos contaminados. Entonces me llamó mucho la atención la existencia de estas bacterias que tú llamas como parásitas al interior del nódulo. ¿Son las mismas que se conocen como los cheaters, que son las que engañan, o no son los mismos? Esa es la primera cosa. Y luego, si ese rhizobio que encontraron al interior del nódulo es del mismo tipo de rhizobio que estaba nodulando, me refiero a que si eran de los mismos rhizobium leguminosarum o era otro rhizobium, para explicar un poco de la interacción y que quizás se complementan para poder llegar a hacer la fijación completa en la leguminosa.
O sea, para la primera pregunta, si son cheater o no, yo siempre tengo un poco cuidado de usar ese término, porque yo sé que hay gente, especialmente los que son como como theoretical biology, son súper quisquillosos en cuándo usar la palabra cheater, porque cheater tiene que ser, o sea, se supone que tiene que ser que la bacteria activamente tiene un mecanismo para engañar a la planta. Y esto podría ser simplemente que la bacteria no ha mutado nada, sino que simplemente no se puede comunicar con la planta porque le falta alguna señal, le falta alguna proteína, pero porque podría ser que esta no es la planta hospedera correcta. Entonces, como un mecanismo activo para engañar a la planta o para usufructuar de la planta, no tenemos evidencia concreta. Por eso evito llamarlas cheater. Eso es para la primera pregunta, pero yo sé que hay mucha gente las llama cheater, que son este tipo de bacterias que son simplemente cheater si es que nodulan a la planta y no fijan nitrógeno. Y respecto a la segunda pregunta, estas dos bacterias que colonizaban, o sea, que se aislaron de este mismo nódulo, la otra bacteria, la segunda, es un mesorrhizobio, y este mesorrhizobio puede nodular y puede fijar nitrógeno sin ningún problema. Entonces, no se necesitan las dos bacterias para fijar nitrógeno. La otra es absolutamente capaz de hacerlo por sí sola. Lo que creemos un poco es que esta bacteria es como, ¿cómo se llama? Como que usufructúa de los nódulos que forma la otra bacteria y se cuela dentro del nódulo. Porque el nódulo es un ambiente súper rico para la bacteria, entonces tiene carbono. Si es que la bacteria puede entrar, va a beneficiarse mucho va a beneficiar mucho. Pero no sabemos cuál es el beneficio para la planta. Vemos que no hay ningún costo. Cuando ponemos las dos bacterias juntas, la planta recibe todo el nitrógeno de la otra bacteria, pero no sabemos si es que hay algún beneficio para la bacteria que está fijando nitrógeno, porque decimos, o sea, está haciendo todo el trabajo de fijar el nitrógeno, ¿para qué necesita la otra bacteria? Y eso también lo estamos investigando un poco, pero los resultados no son tan como claros, así que por eso no quise mostrar, pero lo que vemos es una tendencia a que la otra bacteria, la que no fija, ayuda a la colonización de la raíz. Hay más colonización de la bacteria que fija cuando la ponemos con la que no fija. Porque no sé si se acuerdan de la imagen que mostré, la que no fija coloniza mucho la raíz, coloniza muchísimo, parece que hace mucho biofilm hace muchas biopelículas en la raíz. Entonces creemos que puede estar ayudando a la otra bacteria a colonizar, o tal vez en la etapa de la rizósfera, o la colonización de la rizósfera, ayuda a la que fija nitrógeno, y una vez que la otra más hace el nódulo, la otra se cuela y entra. Pero todavía no tenemos tanta evidencia para eso.
Ok, súper. Muchas gracias, Macarena.
A ver, y si hay alguna última pregunta. Paulina,
Hola.
Adelante.
Hola Macarena, me gustó mucho su presentación.
Gracias.
Yo también trabajo con promotores del crecimiento vegetal. Me gustaría saber si tienes alguna idea qué pasa con los otros microorganismos, bacterias, digamos, que no son relacionados a rhizobium. Si tienen esta misma respuesta a las plantas de formar estos simbiosomas. No sé si algo de eso.
Hay un paper del laboratorio de Simona Radutoy en Aarhus, en Dinamarca, y ella lo que investigó fue que, o sea, lo que vio ella es que otras bacterias que no son fijadoras de nitrógeno pueden entrar al nódulo a través de estas hebras de infección, como que sí, la bacteria fijadora o el simbionte crea esta hebra de infección y las otras cepas pueden entrar por este tubo que hace la planta. Y ella muestra cierta evidencia que la bacteria también entra a la célula, no es solo que se quede en el apoplasto como la mayor parte de todas las bacterias de los endófitos normales que se mantienen en el apoplasto, estas pueden entrar. Lo único que sí es que estas bacterias también eran rhizobios, no eran simbióticos porque no tenían los genes de NOT o NIF o FIX, pero también eran rhizobios. Yo hasta el momento no he visto evidencia de otros que se hayan visto dentro de la célula de la planta, pero eso nos gustaría mucho. O sea, hay un consorcio en Alemania que es en el que estudiamos el microbioma de las plantas, y en el proyecto de mi estudiante de Duncan estamos viendo exactamente eso, o sea, cuál vendría a ser el rol de otros endófitos, de potencialmente endófitos que colonizan el nódulo, y ver si es que pueden entrar a la célula y si es que hay algún tipo de interacción con los rhizobios, por ejemplo, para promover más aún la fijación de nitrógeno. O tal vez algún efecto con el sistema inmune de la planta, o tal vez con adquisición de otros nutrientes, no sé, hierro o otros compuestos, solubilización de fosfato.
Claro. Súper, gracias.
De nada.
Y parece que Mijael tenía una última pregunta, pero no te escuchamos. Tienes que encender tu...
Perdón, disculpen. No, quería hacerte una pregunta un poco más teórica, si tú quieres, pero así como hablabas de estas bacterias que son tan benéficas para la planta, ¿cierto? También hay estas otras bacterias que son parásitas para las plantas, ¿no? Entonces, ¿cuál es la decisión? ¿Cómo la bacteria toma la decisión, digamos? Si irse por el camino benéfico o por el otro, ¿no?
Eso también va por un lado esto de la biología más teórica, donde la gente que estudia la simbiosis teóricamente, lo ven como un espectro que va desde el parasitismo absoluto hasta el mutualismo. Entonces, y hay como un gradiente que se mueve, y hay siempre una especie de, no sé, si quieres, de arms race entre el simbionte y el hospedero, porque el costo de la simbiosis es bastante alto para la bacteria. La bacteria termina, o sea, en algunos casos en la simbiosis la bacteria queda totalmente esclavizada para fijar nitrógeno, incluso la planta no puede reproducirse más. Entonces hay como un incentivo para la planta, en teoría hay un incentivo para la bacteria para engañar a la planta y convertirse en el títer que Carolina mencionaba. Pero la planta, por otro lado, establece muchos niveles de control para que eso no ocurra. Entonces hay como un gradiente que se ve en la naturaleza, que va desde... Pero la bacteria no puede decidir eso totalmente, porque se ha visto en casos que si la bacteria no fija, la planta la mata. O sea, hay controles que no se sabe muy bien cómo funciona, pero hay niveles de control para que las bacterias no escapen, no se conviertan en cheaters. Hay distintos niveles de control por parte de la planta, pero es complejo.
Ok, muchas gracias.
Bueno, muchas gracias a todos que preguntaron. Muchas gracias, Macarena, por excelente coloquio. Y los quería dejar invitados para el próximo coloquio, que va a ser a finales de octubre, que va a estar la doctora Marta Hengst, que es de la Universidad Católica del Norte. Así que también los esperamos el próximo mes en el coloquio de Microbiología de Valparaíso.